Hey everyone, and welcome. We're all used to getting medical reports that are just a bunch of numbers on a page. But today, we're going to look a little closer, because sometimes the biggest clues aren't in the numbers at all, but in the incredible, visual story our own blood is trying to tell us. So, let's dive right in. So, whenever you get a complete blood count, a CBC, you get this report that's just jam-packed with really precise data. And don't get me wrong, our modern automated counters, they're amazing. They tell us the what and the how many of our blood cells, the count, the volume, the concentration. They are truly the backbone of modern hematology. Well, that's the thing, isn't it? Sometimes the numbers look perfectly normal, or maybe they're just a little bit off, but something is still seriously wrong. The numbers give us the facts, sure, but they don't always give us the why. And in medicine, the why is everything. Turns out the real clues are often hiding in plain sight in how the cells actually look. And that is exactly where the peripheral blood smear comes in. Now, this isn't about replacing our amazing tech, it's all about complementing it. This is where the art of medicine really meets the science, using the most sophisticated pattern recognition machine on the planet, the human eye. What's really interesting is that even with all of our high-tech advances, this basic truth just doesn't change. Like the slide says, automation is an incredibly powerful partner, but it complements manual microscopy. It doesn't replace it. It gives us speed and precision, yeah, but it's the skilled eye of a technician that provides the real interpretation. It's kind of like the difference between a printed ECG report and a cardiologist actually listening to your heart. So here's the bottom line. The machine is fantastic at counting. It tells us, yep, you've got anemia. But the human eye, looking at that smear, it can see why. Are the cells too small? Are they literally being ripped to shreds in the bloodstream? Are there actual parasites hiding inside them? That's the qualitative info, the real story that gets you to a diagnosis. Okay, so before we can read this amazing story, we have to prepare the page. And this is where a really simple but incredibly skilled technique turns one single drop of blood into a diagnostic masterpiece. This is the classic wedge slide technique. It looks simple, right? You take a tiny drop of blood, you use another slide as a spreader, and then with a quick, smooth push at about a 30-degree angle, you create this beautiful, feathered edge where the red blood cells lie in a perfect single layer. We call that the zone of morphology, and it's where the cells are ready for their close-up. All right, now for the fun part, the real detective work. When we look into that zone of morphology, we're not just looking for normal, healthy cells. Nope. We're on the hunt for the unusual suspects, cells that have changed their size, their shape, or their color. And every single one of them is a clue. Let's start with the most basic clue, size. Are the red blood cells microcytic, smaller than they should be? That immediately points us toward problems with making hemoglobin, like you'd see in iron deficiency or thalassemia. Or are they macrocytic, way too big? Well, that suggests a problem with how the cells are maturing, which is often linked to things like a B12 or folate deficiency, or even liver disease. Next up, we've got target cells. Look at them. They're a perfect example of how shape tells a story. They get that distinctive bullseye look because the cell membrane is actually too large for the amount of hemoglobin inside. So the extra membrane bunches up right in the middle. When a lab tech sees these, boom, conditions like thalassemia and liver disease jump right to the top of the list. Now, these are really dramatic. Schistocytes aren't just a little misshapen. They are literally fragments of red blood cells. They can look like little helmets or triangles because they've been physically sheared apart just trying to get through damaged blood vessels. Seeing these on a smear is a major, major red flag for some really life-threatening conditions. And here we have one of the most famous shapes in all of medicine, the iconic sickle cell. This shape isn't random at all. This specific crescent or sickle shape is the direct result of a tiny single-point mutation in the hemoglobin gene. It's a true molecular disease, and its signature is written right there on the blood smear for us to see. These are teardrop cells. They look exactly like their name, and believe me, they carry a powerful message. This shape often forms when red blood cells have to literally squeeze their way out of a bone marrow that's been taken over by fibrous tissue or even tumors. They look deformed because they've had such a difficult journey just to get out into the bloodstream. So the clues aren't just about the overall shape or size, but also what's hiding inside the cell. These little dots and bodies are called inclusions. Howell jolly bodies, for instance, are leftover bits of DNA. A healthy spleen would normally yank those right out. So seeing them tells us the spleen isn't working right. Or basophilic stippling. That can be a classic sign of lead poisoning. It's incredible. Okay, so far, we've been looking at problems with the cells themselves. But what about problems that are literally living in the cells? The peripheral smear is also our frontline defense for finding microscopic invaders. And you really can't overstate this point. When it comes to diagnosing malaria, one of the most widespread infectious diseases on Earth, the blood smear isn't just a good tool. It is, and remains, the absolute gold standard test. All the rapid tests, all the new technologies, they're ultimately confirmed by a trained human eye looking through a microscope. And diagnosing malaria is actually a two-step process. First, a thick film is examined. In this one, the red cells are basically destroyed to concentrate everything, making it way easier to spot any parasites, especially if there aren't many. Then, once a parasite is found, the technician switches over to the thin film, just like the ones we've been looking at, to identify the exact species, which is absolutely critical for treatment. And telling the species apart is all about looking for these subtle visual clues. For Plasmodium falciparum, which is the most dangerous kind, you might see multiple tiny ring forms inside a single cell. For P. vivax, on the other hand, you'll see that the infected red blood cell itself looks bloated and enlarged, and the parasite inside looks more like a little amoeba. It's fascinatingly detailed work. So we have all of these visual clues: size, shape, things inside the cell, even parasites. But how do we turn all these observations into a report that's actually useful to a doctor? I mean, we can't just write down, saw some weird-looking cells. We have to quantify it. And that's where grading comes in. It's not random. The observations are systematically counted and then graded on a scale, usually from 1+ to 3 or 4+. So seeing maybe 1 to 5 teardrop cells over 10 different views might be graded as 1+ or slight. But seeing more than 15? That's marked, and it points to a much more serious problem. This whole system adds a vital, semi-quantitative layer to the art of reading a smear. And really, it all comes back to this one idea. The automated counter, the machine, it gives us the headline, anemia present. But the peripheral smear, that gives us the full story. It gives us the characters, the plot twists, and all the context that leads to a real understanding of what's going on with a patient. And it just leaves you wondering, doesn't it? For more than 100 years, we've been learning the language of our blood cells just by looking at them. It makes you think, as our tools get even more powerful, what other incredible, detailed biological stories hidden deep inside our own bodies are we on the verge of finally being able to read? Thanks for tuning in.